Okay, so thank you again all so much for having me here. I think it's safe to say that all of us in this room value biodiversity. Our work as researchers, as educators, as advocates really highlights why biodiversity matters and why we should protect it. However, I also want to argue that this is not represented in the types of people, scholarships, and value systems that we engage with in our daily work. So I would like to invite you to think about this as we go through the seminar, and to think about ways that you could continue to increase the diversity of viewpoints and researchers that you engage with. I also want to take a moment to acknowledge that the entirety of my work has happened in Indigenous land and without the consent of these land's rightful stewards. Well, I hope that you learn a lot, or at least one thing from my seminar. I also know that a lot of the knowledge that I will share with you probably already existed and was known by Indigenous scholars and elders in the Americas and beyond. So with the effort of sharing Indigenous knowledge that I've really gained a lot from, I want to share a reflection from Dr. Ren Walker Robbins, who is an Indigenous scholar and a professor at Salish Kootenai College, and whom I had the privilege of learning from during my time at Minnesota. And the thought goes like this. So if we look out of a window in our homes, into our yards, it would be hard to think that having just one grass species happened by accident, that would just naturally by accident happen. We spend a lot of effort making our lawns to be just one species. However, when we think about natural communities like grasslands, they would tend to have lots of different species. However, they are starting to look more like this picture on the right than they do like the picture on the left. We agree that this loss of diversity globally is because of our actions. It's because of the things that we're doing or not doing to protect diversity. At the same time, if we think about who's represented in this room, whose scholarship we read, value, cite, it probably looks a little less diverse than perhaps it ought to be. And I want to argue that that really matters. So I want to start by sharing some recent results from a biodiversity expert survey that I had the privilege of participating in. So Forrest Isbell, my PhD advisor, led this huge effort to survey over 3,000 biodiversity experts from 113 countries. And one of the questions we asked them was, what should be the top priorities for spending in conservation? And we got very different answers depending who you asked. So experts that came from high-income countries thought that we should spend most of our conservation money acquiring and protecting areas that are not currently protected. So buying land, protecting it. Whereas if we ask experts that came from lower middle income countries, their thought was that we should spend more money in both monitoring and managing both protected and unprotected areas. So you can already see that really putting effort into being more inclusive really changes the types of questions and the types of answers that we come up with. In my work, I think about these questions in natural systems. So for the rest of the talk, we'll be mostly talking about plant diversity. But generally, work from many colleagues has shown that communities that have high diversity tend to have many benefits to society. So for example, food production, nutrient and water cycling, pollination services. We also know that different perturbations tend to lower diversity of these communities, and that also reduces the benefits that people get from nature. And the big question that I'm excited about is thinking, as we start relaxing these pressures on natural systems, will we see that those systems readily recover into those high diversity, high ecosystem service provisioning systems, or have we perhaps pushed it to a point where we're stuck with a lower diversity, lower provisioning of benefits to society? And the way that I do this is by using a variety of techniques and variety of different approaches. So I like working with long-term data from both experiments and observational data sets. I like working with lots of different collaborators, which really changes the types of questions that I get to ask. And I like using those tools and systems to think about the mechanisms of both spatial and temporal change of plant communities, with the objective of thinking of what could be potential good solutions for long-term conservation outcomes. Today, I will share with you just 3 examples of how these different tools inform my research, and just give examples of some of the type of work that I do. So first, let's talk about some experimental evidence for recovery of diversity after different perturbations. And I just want to start acknowledging that most of what I will share with you, most of my work happens within the long-term ecological research network, which is national and also global series of sites that just have really, really long-term experimental manipulations, and we tend to be a very collaborative network. For this part of the talk, I will specifically talk about work that I did at Cedar Creek Ecosystem Science Reserve in central Minnesota. And the reason why Cedar Creek is such a great place to ask questions about diversity recovery after perturbations is that there is a whole series of long-term experiments that have added nutrients and then stopped adding nutrients. So we have this long-term perturbation followed by a long-term period of recovery after it. And if we were to fly above the fields at this field station, you might notice that there's little squares that look slightly different. They are different because the plant communities have changed based on the rates of nutrients that we have added to those plots. So even just from the picture, you can already tell we've changed community composition in those plots. These experiments it's a series of experiments, and they were set up by Dave Tillman in the 1980s, and they've been ongoing ever since. And the general story of what happened in these experiments is that they used to be all agricultural fields, so they were plowed at some point. But long enough had passed that by the time the experiment started in the 80s, the plots had high diversity, 12, 15 species per meter square, and soil nitrate was fairly low. The soils in Minnesota are very sandy, so very nutrient limited. As we added nitrogen and other nutrients, we saw that species diversity started to decline. And when rates of nutrient addition were very high, within 10 years, we went from having communities with 15 species to having maybe 1 and 2. And the identity of these species also changing. So going from having mostly native dominated communities to having communities dominated by 2 exotic species. At this point, Dave and collaborators decided that they were fairly convinced that if you add nutrients to these old fields, you will lose diversity. So they thought, let's think about something else. Let's think about whether these communities would readily recover if we stop adding nutrients. And I would like to claim that I had anything to do with it, but I was born in 1992, so nothing to do with it. I'm just very lucky to be part of a network that has people working on these experiments through generations. So what happened is that they stopped fertilizing half of the replicate plots in the experiment, and if we went to sample them, now you would see that the ones that receive high rates of nutrient addition still have just a few species that are cycling in abundance, but still just very few exotic species. So this, I would argue, is evidence of a discontinuous transition in biodiversity in response to nutrient enrichment. And if we went to the plots, this is an example of a plot that received medium rates of nutrient enrichment. So you can see that if you didn't add a whole lot of nutrients, you do have the ability to recover. So this is a picture from a class I used to teach in Minnesota, where you can see that there are a few species coming back into these plots, nowhere near as diverse or as productive as perhaps the back of the field that you can see in the picture. And the reason why we might have these persistent changes, there could be many mechanisms. So if we think about the types of communities that we find in these plots, they're very different. So we have, for the plots that didn't have a lot of nutrients added or didn't have nutrients added, we have diverse communities with deep root systems. For the ones that had high rates of nutrient addition, just a few species, mostly rhizome spreading grasses, so very shallow root systems. The types of microbes that are present in the plots are also different. perhaps the associations with the microbes have changed as well. The accumulation of litter is vastly different. So if we were to go into the plots and just see how much litter there were, you would be able to easily put your hand through in the plots that are mostly native dominated versus in the ones that are exotic dominated. It's a very thick mat. It's wet, there's no light going through, very different light environment. And previous work has also shown that there are persistent changes in the rates of nutrient recycling in these plots. That altogether might change the ability of species to disperse back into those plots and be able to reestablish even after nutrient enrichment has decreased and soil nutrient levels are lower again. So a lot of my work in Minnesota was to try to figure out mechanisms by which we could help recover those species. So I tested the usual suspects of recruitment limitation, perhaps persistence of high nitrogen levels. So carbon addition is often used to temporarily capture some nitrogen and allow species to establish. Seed addition reduces recruitment limitation, removing litter to increase the amount of light penetrating to the soil layer, and then prescribed burning that changes both the amount of light, but also the forms of nitrogen in the plots. So first, let's look at results from prescribed burning. So just to remind you, the experiment started in the 1980s. Plots that received high levels of nitrogen and other nutrients had low diversity. And then what we learned from looking at temporal data of the first 10 years of the experiment is that diversity is declining everywhere. No matter if you are just a control plot, we're us experimentally not doing anything, diversity is declining. We also know that decline in species gets accelerated when we add nutrients. So the loss of species is much faster when we add nutrients. So then after 10 years of just adding nutrients, what happened in this experiment, instead of stopping fertilization, was prescribed burning. So the plots are burned 2 out of every 3 years. So it's fairly high fire frequency. And that was also in the 90s, and half of the replicate plots are burned. And what we find from this time period is that burning does slow down the rates of species losses. So even though nitrogen accelerates it, burning can help us decrease that rate of species losses. And then around the time where I showed up in Minnesota, we decided to make this experiment, overcomplicated experiment, a little more complicated, and we stopped fertilizing half of each replicate plot. So now we have a fully crossed burning and cessation and a diversity gradient. So I'll show you results from this end period of the experiment. I have grouped the nitrogen addition treatments into no nitrogen added, low, medium, or high levels. I do want to point out that the experiment started in the 80s. We're not necessarily concerned with the questions I had. So even though I'm focusing on the rates of nitrogen, there's also all other nutrients being added. So pH dynamics are different than you would expect if you were just adding nitrogen by itself. And then on the y-axis, we're going to look at richness. And if you are curious about what those the exact amount of nitrogen is, the numbers are there. And I just want to remind you that this is data after 28 years of experimental burning and then 5 years of cessation since cessation of N addition. And what we find for plots that receive no or low levels of nitrogen is that burning increases richness. So if we burn, we can get more species remaining in those plots than if we don't burn. When we have medium rates of nutrient addition, it starts getting interesting that you need both to burn and to stop adding nutrients in order to get that species diversity back. And then once you look at plots that have high levels of nitrogen added, then we don't actually see that big an increase of either burning or cessation or both of them together. So that would indicate that, at least for the moment, that treatment is not really helping us increase diversity. And if you'd like to see it visually, I have some pictures. So here are 3 plots next to each other that show what a control plot looks like. So at this time of year, when I took the picture, it's mostly dominated by little blue stem, but there's also all sorts of other species in there. Then the plot in the middle is a plot that received continuous fertilization, so still being fertilized. You might see that it's mostly a mat, but it's very green. And then closer to us is a plot that had been fertilized and stopped. It still looks like the same species, it just looks a little less green, but still same thing happening. I do have a lot of data, so even though I don't have time to show you, the effects do increase over time. So it is possible that with more time, we might start seeing recovery of those plots. So let's think about other potential management strategies. We know that not all ecosystems are like fire, some are fire sensitive. So perhaps we could think about other mechanisms that might help us increase diversity. So for this experiment, I had the privilege of collaborating with Chris Clark, who's a scientist at EPA, Carmen Watkins, who was an undergrad student at Minnesota and now is a graduate student in Oregon, and then with Forrest. And the experiment is very similar. So started in the 1980s, fertilization now at just 1 rate. So the medium rates of nutrient addition, where we had those interesting dynamics with fire and cessation. Fertilization stopped for the whole experiment in the 1990s. And then in 2004, when Chris was doing his PhD, he thought, well, how about we do some experimental manipulations and monitor what happens in the short term for recovery of species diversity. And he then finished, and the experiment was left there. And no one visited it again until 2017, when I was there and I was excited to find it and figure out what happened in the long term. So just to remind you, the treatments are seed addition, which reduces recruitment limitation. We added 10 species at 0.5 gram per meter square each. We removed litter manually and then added carbon. And all of the, it's a full factorial design. So there's all combinations, 7 replicates each. And then the treatments were applied once in 2004. So we're looking at long-term outcomes 13 years after a one-time intervention. And as I mentioned, the maps, I was lucky to have this experiment happen when there was a transition from paper records to digital records. So there was no clue of where the experiment was. So it looked just like this. I had to map with Carmen a lot of the experiments. We found lots of old things. You should clean up your stuff from the field. Otherwise, someone else will find it in 15 years. Eventually, we did find it, and this is my brother, who was very helpful and continues to help me do a lot of my field work. So we're able to find and sample the experiment. And if you're not familiar with the way that at least grassland community colleges gather data, essentially you clip a strip of biomass and then you take it back to the lab. You identify the different species that you have, and then you separate them, bag them, which allows you to look at the amount of biomass that each species is contributing to the total biomass production. So for these results, I will show you the effect on species richness on the x-axis, and then on the y-axis, the effect on above ground biomass. And it's all in reference to control plots. So remember, those are plots that were previously fertilized, but there was no management treatment happening there. So if you are a land manager, out of all those combinations, you would hope that your treatments would land you somewhere in this top right corner where you have more species and perhaps more above ground biomass, more production. And what we find, I'm showing you all, but the main thing is that all of those warm colors are treatments that had seed addition. So seed addition was the only treatment that increased both richness and above ground biomass production for the whole experiment. you can see the other treatments, even though in early years of the experiment, litter removal seemed to be very important. 13 years later, the main thing is whether or not you added seeds. And one of the more exciting things is that seed addition decreased the biomass of those 2 exotic species that I had been mentioning. So quackgrass biomass decreased by around half. And then Kentucky bluegrass, not as big a reduction, but still significant reduction in the biomass of that species. At this point, you might say, okay, cool, you added a bunch of seeds, you replaced everything that was there. Shocking. And you're partially right. So a lot of the increase in biomass is due to the persistence of the species that we planted. However, even though we decreased the abundance of those exotic species, we did not decrease the abundance of the already present native species. So adding seeds did its job. It allowed for native species to persist, the planted species to remain as well, and it reduced the abundance of those unwanted species in our plots. So if you ask, can we get back from those communities with low diversity, low productivity, back into those more desirable high diversity, high ecosystem service provisioning types of environments? And I would say that you can. So when we put results from these 2 experiments together, you can see that both seed addition and burning are good strategies to increase diversity in the long term. Now I would like to share with you some of my work thinking about temporal and spatial mechanisms of ecosystem transition. So this is some of the work that I was doing as a postdoc and continuing to do right now. So I'm really excited about thinking about both time and space and how considering both of those aspects might change how we manage communities in the long term. So thinking not just of what works now, but what might work in the future, and whether the map of where things might work is shifting as well. So during my postdoc, I worked with Annie Chung at University of Georgia. Annie Chung is one of your own. She went to undergrad here. And then did her graduate work at Seviera LTR in New Mexico. So we were working together to think about this different type of transition, but also an important shift in community composition. So thinking about changes from a desert grassland into a shrubland. So what's been happening in Seviera is that there's been an increase in the abundance of creosote bush, and there's this really amazing ecotone transition that we went and worked at. So the questions were, how is creosote able to increase abundance and sort of start going into places where black grama, which is the most dominant grass in those places, is. And if you've never visited a desert grassland, which I hadn't before my postdoc, I just think you should look at a picture. It's so different. We often think of these systems as being very similar, but you can see that there's at most 50% cover. So if you tend to work in Missouri or places that have a lot of rainfall, this might be surprising to you. It was definitely surprising to me. We were working to understand whether changes in the abundance of creosote bush could be mediated by plant soil feedbacks. So we established an experiment where we identified sort of target individuals for both of the species, and then we had a grid going out of those 2 species where we dug holes and had either live soil or sterile soil where we planted seeds for both of these species. So that way we could look at both biotic and abiotic mechanisms of spread. And what that looked like at the end is that we had a lot of little cylinders with the special type of filter paper that held the soil. And then you can see lots of different toothpicks. So we had like thousands and thousands of seeds out in the field. Annie and the rest of the group were just harvesting those plants last week. So I unfortunately don't have the data to show you, but I can tell you about what we're doing with that data. So during my postdoc work, I was working to create a model where we could start thinking about how that data could inform mechanisms of community split through plant soil feedbacks. So I developed this individual-based model where we could start changing the area and the time by which each species influence the cells around it. So thinking the soil around it. So for example, we could think about the neighborhood of competition and plant soil feedbacks as being different size. Which one should be bigger? We could argue that, but the beauty is that it's a model. So we could do both. We also thought of whether the spatial extent of plant soil feedbacks would decay as you got further away from the plant. So for example, when we were digging our holes to set up our experiment, we could tell that the amount of roots decreased as we got further away from the plant. So it would make sense that there would be a similar effect on the soil on the influence on the soil microbes near those plants. And then also the legacies of plant soil feedbacks. So after a plant dies, for how long is the effect on the soil still apparent? And a lot of people have done work thinking about how plant soil feedbacks build over time. So as you have an invasion front or as you have species coming into a place, how fast do they accumulate either mutualists or pathogens that affect their growth? But we had a harder time trying to figure out how long would the strength of plant soil feedbacks last. So thankfully, I had a data set from, from my work in Minnesota where this happened. So as you might remember, we had native communities dominated by little bluestem that became invaded by quackgrass and exotic species. So as you might expect, if we just looked at the relative biomass of little bluestem over time, it changed based on the treatment. So we lost or had reductions in the abundance of little bluestem in each plot at different times due to how much nutrients we added. And is it a perfect way to test the legacy of plant soil feedbacks? Maybe not, but this is what we have. So what we did is that we calculated for each plot in the field the area under the curve for the relative biomass of little bluestem. So essentially like an integrated measure of how much little bluestem was there and when did it go away. And then we had this greenhouse experiment where we grew little bluestem with soil from all of the plots in the field. So essentially we had a measure of what the responsive growth of little bluestem seedlings would be based on how long little bluestem had gone away from the plots or how abundant it had been in the long history of the experiment. Again, it changed over time, so we had a measure for each plot, several replicates, and what we found was that perhaps it's not that long. So we do see a negative response. So if we look at area under the curve here for little bluestem, so an abundance metric for the grass, and then the little bluestem biomass in the greenhouse, we do see that the more little bluestem was present in the plots in the field, the less it grew in the greenhouse, but it's not a very strong relationship. So probably not a major driver of little bluestem growth, at least in this experiment. But at least it was our first step at trying to get data for this different system. And in the next couple of years, we will have actual data that is in an experiment that is actually set up to answer that question. Lastly, I want to share with you how I work with diverse groups of collaborators to think about both theory and synthesis of community shifts. So taking all of this data that we've gathered as a network and then asking cool questions about it. So this is work that's supported by the Long-Term Ecological Research Network and also NC, the National Center for Ecological Analysis and Synthesis in Santa Barbara. And I had the privilege of learning how to do this. As a graduate student, I was invited to participated in a working group at MC's where we were thinking about species synchrony. I don't have much time to tell you all about the work that we did, but there are some papers that recently came out that you should definitely check out. So Lauren Shoemaker and Lauren Hallett led a paper thinking about community synchrony and how it varies by time scale. So thinking about variation in species in short time frames, so 2 or 3 years versus 10-year time scales, and how those might contribute to stability. John Walter led a paper looking at how community properties also vary synchronously over time and space. So for example, thinking about species richness and how that itself can vary spatially in a synchronous way. So even though we might think, you know, a heterogeneous environment, there will be differences that will sort of even out, there actually is synchrony on some of those community metrics. And then Lauren Shepard is leading a paper that is separating what drives these fluctuations. So looking at non-independence of species fluctuations and how that increases variability. So similarly, thinking of species are varying and co-varying in different ways. And when we think of communities that are 15, 20, 30 species, there is some redundancy on how they're varying. So some correlation on how they're increasing and decreasing in abundance. So you should definitely check it out. I'm sorry I can't go into more detail, but I want to share with you some of the work that we're doing in the new working group. So after that, as I was starting to think of what I wanted my research program to be, I've always been interested about thinking of multiple perturbations. So we know that communities and ecosystems are not just facing nutrient enrichment or just changes in fire frequencies or changes in how much carbon dioxide is out there. All of it is happening at the same time. Also, they have ever increasing climate variability. So changes in the magnitude and frequency of big storms, heat waves. And I just thought this was a really cool question, and that we have experiments now that are explicitly testing some of these questions. So for example, at Minnesota, I helped set up one of the DroughtNet sites. So DroughtNet is a distributed experiment, global experiment that is imposing a 1 in 100-year drought on different plant communities. So I set up one of these in Minnesota. And the exciting thing was that I was able to set one up on top of another long-term experiment. So it was an experiment that had manipulated irrigation, so added rainfall, and also fertilization, so adding nutrients. So for 10 years, plots had been irrigated, fertilized, or irrigated and fertilized. And then on top of that, I was able to impose my experimental drought. So then we can ask really cool questions about whether there are legacies of rainfall, the history of rainfall, on how a community would respond to an extreme event. And again, that's a paper I'm working on now, but I will share with you some preliminary results. So if we look at plots group based on their previous irrigation, so if they were not previously irrigated, that's just ambient rainfall. And then if they were previously irrigated, then that means we added water with rain events. And then we're going to look at the change in above ground biomass. And then rainfall reduction, the ones that are open circles don't have a drought, the ones that have closed red circles had a drought. So first, if we just look at plots that had not been previously irrigated, so just ambient rainfall, we see that when we impose a drought, biomass declines, so that is perhaps not surprising. Declines by a lot. That happens. If you don't water your plants, they're going to grow less. But then when we have plots that had previous history of irrigation, we still see a decline, but it's nowhere near as big as when we have not irrigated. So I think this is evidence that the history of rainfall does change drought responses. And then thinking back to all the many experiments like this, DroughtNet and beyond, that are manipulating both different global change factors, but also explicitly manipulating climate events, I just thought we should ask questions about this. So with Amy, I wrote a proposal for a synthesis working group at NCs, where we're asking questions about the role of multiple perturbations and increased climate variability in driving these difficult to reverse compositional shifts and also the functions and identity of the members of the community. So we have this amazing group of collaborators. I feel very excited that we have colleagues that are of all career stages. We have people that are theoreticians, but also people that work at USDA and do more applied work. So we have a real diversity of career stages, expertise, and we have had one meeting in online, and we have our first in-person meeting. Like, we're all finally going to meet in person next week. So I'll share with you a little bit of what we're doing. So again, taking data from experiments across the LTER, but also beyond, to ask questions about how these different perturbations change different aspects of community. So for example, we have groups within our working group that are looking at the effects of perturbations just on community variability. So how are just these global change drivers changing the way communities vary over time and space? We have a group of people looking at effects of climate variability on community variability. So thinking about either places that have experienced very variable climate or where we've manipulated climate, does that result also in more variable communities? A group that is thinking broadly about ecosystem transitions and whether or not we find evidence for them. And then we have another group thinking about taxon-specific responses and explicitly thinking about how do we think of rarity and dominance in communities when we start thinking about both temporal and spatial presence of these species. So I'm leading the effort to think about the broad ecosystem transitions question. And we decided that even though we have a lot of data that we've already identified, because it's the places where we work, we want to ask how often do we actually find evidence of these difficult to reverse transitions and be a little bit more inclusive and broad about what we are considering. So we're building on this review from Schroeder from 2005 that looked at essentially reviewed the literature to look for experimental evidence of ecosystem transitions and recovery. So essentially, we are in the process of doing a repeatable search of the literature. There's so many more papers. In their paper, they're reviewed in the magnitude of dozens of papers they found. We found now thousands of papers. But essentially looking at how environmental parameters, so those global change factors or nutrients or anything that people manipulate, grazing pressure, for example, is a big one, might drive a state variable. So that could be diversity or productivity, or it could be different for different systems. And essentially we're looking for experiments or observational data sets that test for either discontinuity. So places where we have pushed the system, say, for example, the fertilization experiment I shared with you, and then we've stopped fertilizing it. So the environmental parameter, we pushed it one way, we pushed it back. Do we have that discontinuity that we're still in this same state? Test of no recovery. So when we just push the system off a cliff, that's another case. And then test of divergence. So if you have different initial conditions, will that result in very different outcomes long term? As I mentioned, we found over 3,000 papers, and we did a first screening of titles and abstracts just to make sure it was a field experiment and had the type of data that we can analyze altogether. We do have a bias for terrestrial herbaceous communities. We did consider doing it broader, but then that got us into the dozens of thousands of papers. So we decided we didn't quite have capacity for that. But right now we found 400 papers that we're processing further Because in many cases, these experiments are very long-term, so there might be repeats of the same experiment showing up in different publications. So what we're doing next week is that we're going to take the experimental data from those published papers. We're going to go find it. Most of them are readily available online, and then we're going to find the climate data for those places. So even if in the paper they don't report climate data, we can go find it. That is readily available. We can go with coordinates of where the experiment happened and the time frame. We can find climate, and then also if there are other drivers. So for example, rates of nutrient deposition for those time periods. And then we're going to ask questions about whether extreme climate might be promoting persistent changes in communities. So just to go back to the example that we've been talking about, changing from a native to an exotic community. If we were thinking, say, if we wanted to put this in an equation or in a graphical way, about on the y-axis, short-term changes on invasive species cover. So if we have an increase, that means there's more, decrease, there's fewer. And if we look at how that changes based on how much invasive species cover is there, we might expect that under historical climate, when there are few invasive species, there's perhaps not enough of them to sustain short-term increase in their abundance. So maybe a few will come in, but overall they're not gonna invade your plots. However, if there might be a threshold after which, if you have lots of these species coming in, they successfully invaded the plot, and then you would end up with this bistable system where you would have either plots that are, or places that have very little invasive species cover, or places that have lots of invasive species cover. So our big question is, if this is what we see under historical climate and what we would expect, is it possible that extreme climate events make it more likely that we have systems with just one stable state that is more likely to just be in lots of invasive species? And again, just one example that we're familiar with. We can do this both with that readily available data online or using our experiments. And we're hoping that this data product that we're assembling will help us think about this question, but also be a good resource for other people that might have similar questions. And again, these are the types of work that we're doing, and this is just to make a little plug for myself. So I just started at SLU. I started in August, and we are a very small lab at the moment. I have one graduate student, but I am recruiting a PhD student, a postdoc, and a postbac student. So if you love grasslands or are excited about the types of questions that we're asking in the lab, please contact me. Again, we're a small group. Eileen is my current student, if you want to contact her to chat about how our work is going on. But we are essentially very excited to work with people interested in all sorts of plant community and ecosystem level questions. So if this is exciting, please reach out to me or let your students know. So hopefully, just to wrap up, hopefully I've convinced you that long-term data is super valuable. It's so important that we keep funding and maintaining these long-term experiments, that working collaboratively, even though sometimes it is very challenging, it's worth it. It's so much better to think broadly and try to be inclusive in who you work with. And that together can lead to asking more interesting and perhaps difficult to answer questions about mechanisms of spatial and temporal change that I'm hoping will lead eventually to changing the way we think about conservation efforts and lead to better conservation outcomes in the future. With that, I just want to thank all of my sources of support, and then my collaborators from the working group and beyond. And thank you so much for listening.
All right, thank you, Cristy. We have time for questions.
Thank you. That was a great talk. I was wondering if you could talk more about how you did the seed addition. So like, how did you pick which species to add? When did you add them? And that kind of stuff.
Yeah, so I didn't personally do it. This was Chris in 2004, so 13 years before I even showed up. I was a kid. But the way he picked the species was 10 native species that were present in the field. And then he hand casted, like he just measured, like weighted how many would be like 0.5 gram per meter square for each of the species, but they're all native, present in the field. Yeah.
For people on Zoom.
Awesome talk. That got me really excited. I think it's awesome how many long-term experiments you've done, but I have a question about the burning specifically. Just because I'm interested in knowing the biological mechanism that you think is happening when the burning is added. And especially when you see that there might be a decrease in nitrogen. So it, or like even with the nitrogen, potentially what is happening with the burning? Like, are you changing the carbon ratio with the nitrogen, or maybe you're changing the microbes that are there? Yeah, just curious.
Yeah, it could be all sorts of things. So it could be, there's evidence that light limitation is also a driver of like reinforced low diversity not in this particular site, but elsewhere. So by burning, you remove all of the biomass that was there. So you immediately have more light. You both change the amount of nitrogen. So some of the nitrogen in the biomass, like in the litter and the plants, does get volatilized, but then also some of it falls back as ashes. So you have suddenly lots of light and then essentially ashes that are sort of like fertilizer, like good available nutrients. And then just given my bias for the importance of soil microbes, a lot of the spores of plant neutralists like mycorrhizal fungi, they often germinate when you have fire. So these ecosystems evolve to have like fire, they're driven by fire, like their fire is an important way to maintain them. So it could be a mix of all of that.
Yeah, thanks, Cristy. That was a great talk.
Thank you.
It was really neat to see that the seed addition or dispersal increased the total above ground biomass. I'm also curious about the mechanisms there. Is it just that the communities are unsaturated with species, or would you even be willing to go so far to say that it's increasing niche complementarity or like a sampling effect on sort of total productivity?
Yeah, so that was really surprising. So our assumptions, at least at Cedar Creek, generally, like when I showed up and I asked, like, does the seed matter? They were like, seeds are dispersed everywhere. You know, most of the seeds are wind spread. Sometimes plants die, like fall off in the winter into the plots. So you would think that seeds are getting there, but apparently not. So there probably is like an aspect of like spatial heterogeneity. The field is patchy, so there is probably like having those seeds go in is increasing the ability of these species to fill up those niches. And if you get in, it's somewhat debated. I don't know that I trust the numbers of seeds. I just visited Lauren Sullivan's lab at Mizzou, and we're talking a lot about like seed rain amount, but based on what I could read on the literature from seed rain in Cedar Creek, this would be about intermediate dispersal. So it does agree with theory of like you would have like better sorting of species in a heterogeneous environment at these like medium dispersal rates. So yeah, there's not enough seeds coming in, probably, is what I would think. Yeah, great question.
We have a question from YouTube. This is from John Birmingham Jr. Your comment about greater leaf litter in the nitrogen addition plots early in your talk raises a question. Has anyone looked at earthworm density in the plots?
No. that I know of. There are some earthworms, there's not a whole lot of them. I think it might be a driver. I think part of why people haven't looked at earth invertebrates are like soil invertebrates is that the plots are not too big. They're like 4 by 4 meters. So to get or like you would have to dig up the plots, and I don't think they would let me. So it is very possible. It's just not a very.
It's an important issue, though.
It's an important issue, yeah. And I mean, in other places in Minnesota, earthworms definitely drive big changes in communities.
Right, thank you. I was just thinking back that it had been left fallow since 1934. And if it had sat from 34 to 80 something in Missouri, it would be covered with woodies. So was it mowed before then? And was it mowed during? Were the forage and other things removed by mowing, either before or during?
I'm not sure what they did in the, like, before the 80s. But I don't think it was ever mowed. The field is not burned either. Like, at least since the 80s, never burned. There are other fields that are older in the property that are becoming encroached with woodies. So that process is also happening, but just not quite yet, at least in these fields. So there's full chronosequence also of old fields, and yeah, that process is definitely happening.
Hey, thanks for the talk. Because you have replicate sites you have replicates, I think in some cases for, how do the communities respond to addition of nutrients? And you're getting species diversity loss in some cases, yeah.
Yeah.
So I was wondering, did you look across replicates at all to see if similar species were lost and if they were lost in similar orders?
Sort of. I tried to do when we were working with the Synchrony working group. I looked at some data based on so that experiment is replicated in 4 different fields across Cedar Creek, and there are correlations. So if you look at a matrix of like what species are increasing or decreasing in abundance, there are clusters of species that increase together, decrease together. So that's a great question. So that's temporally, and then spatially, not me, but a fellow graduate student, George Fury, did of a neighbor experiment to where I showed you data from. He looked at spatial patterns of diversity, and similar to what I was thinking for the seeds, there is different identities of species in different parts of the field. So the field is heterogeneous, and different species are at different places.
Thanks for the talk. I have a question about, I guess, seed bank dynamics there. Like, is there a possibility of seed being dormant in the soil for over multiple years? I'm thinking about like resilience towards like bouncing back of the dynamics between different treatments.
Yeah, that is possible. I didn't have an opportunity to look at seed banks. I graduated and left, but now that I have a more permanent position, I'm hoping that we can go back and ask some of those questions. So one of the other data sets I'm working on is that in addition to the one-time seed addition, there was another second addition of 4 times the amount. So looking at whether now there's saturation of like what's coming into the plots. So I'm hoping that soon, maybe this coming field season, we'll be looking back at those plots and perhaps doing some seed bank studies. But yeah, we haven't looked at the seed bank. It's very possible. Mm-hmm.
Great. Well, a question that I have is, I'm really interested in the potential of using long-term data from the LTER for various ecological questions. Do you know whether the LTER data are publicly available prior to publication? And if so, how you can find them?
Yes, so you can find it a couple different ways. You can go to the individual sites. The NSF guidance is that 3 years after sampling, data becomes available online. There might be a few exceptions, like if it's a student's dissertation and they need a little bit more time, but generally 3 years after collection, data is available. So you can either go to like usual suspect data portals like Data One or EDI, and you can find a lot of those data sets. You can also go to the LTR network website, and from there you can find all of the individual sites' websites. Often they also host their data sets there, and you can find metadata. You can find, for example, for these experiments that I showed you, you can find spatial maps of where the sampling came from. So like, of course, they don't sample the clip strip at the same place every year. So there's maps of that. But yeah, it's all there. And they usually have emails of who to contact if you have questions about it. And it is a good idea to ask questions about it. So for example, there might be things that happen that are not clear in metadata. So when we were looking at synchrony dynamics, for example, some of the experiments were fenced, and then fences became really difficult to maintain. So then they removed them. So we captured that in our synchronous dynamic, like there was a synchronous increase. I think something happened and people were like, is that a thing? Like, did we find something? What is a climate driver? And it's not climate driver, it's just the fence. So it's a good idea to always talk to the person that might know a little bit about the data. But yeah, the data is just there. You can find it. And then also data products and packages from previous synthesis working groups, those are available through there too. So yeah, you should tap into those.
Great, thank you. Any final questions? Okay, well, please again, join us all for the reception outside at the bottom of Bear Hall, and join me once again in thanking Cristy. Great.
Thank you.
Yeah, need to hear about